This chapter focuses on amino acids, protein structures, and enzymes and how those work. And ultimately, what's going on with proteins is that we have little groups of molecules called amino acids. And those amino acids fit together and end to end and create these long chains. And they're able to assemble and organize themselves into specific structures. And their structure, I have an example of a few different structures, and these are proteins that bind to mRNA. And you can see sometimes they're formed as dimers, or they'll have all of these different types of shapes that we'll talk about in terms of these organizations of themselves so that they can recognize specific RNA molecules. And this is going to be an important part of this idea, is that the structure of the protein tells us something about its function. So how these things are organized in space will dictate what they do. And you can kind of see over here that we have these little structures of, in green is shown where the RNA is going to be, and then in purple is the protein that's surrounding it. So these are important things. We'll talk about how they organize themselves and what they are made up of and how this all works to get a better idea of how they're going to work as enzymes. That's what we're going to focus on really in this chapter, in terms of their functions and how these then are related to later on in terms of biochemistry and how we generate energy and utilize enzymes for biochemical processes. This chapter we're going to really focus mostly on enzymes, but there are different types of proteins that have different types of functions. The enzymes are really important in terms of breaking down different biomolecules and building some of them up and metabolic processes. So that's what we're going to really focus on, since chapter 20 is really about biochemical energy. But there are some other ones that I do want to talk about as well. So there are enzymes, class of enzymes that we'll talk about in a little more detail. And there are also structural proteins. So these proteins can help to organize cellular structure and keep the shape of our cells and attach to other cells and create those attachment points to create different types of structures within some types of the organelles, like flagella, that help different types of cells move around. That these are some of those types of proteins: actins, microtubules, and different filament proteins that provide kind of that highway for things to travel within the cell and to keep those cellular shapes. So these are some examples. There's also things like keratin that create hair and horns for certain animals, and so these are all of the kind of important structural types of proteins. We have transport proteins, things that move different components around within our cells and within our bodies. An example, serum albumin carries lipids within our bodies. Hemoglobin is going to be carrying oxygen and different iron heme molecules as well. Help different proteins crossing membranes, for example, would be a different type of transport protein. So these are all things that different kind of classes of proteins. We have motor proteins as well: myosin heavy chain, kinesin, dynein. These are all different types of proteins that are in our cells and help things move and contract. So myosin is a protein that has these kind of little hands on them, and then when they get energy, they'll kind of contract, and that's what helps our muscles twitch and move. Kinesin's a microtubule-based transport protein that will kind of climb on some of those structural proteins and move things along those kind of highways and structural components within the cells. We have storage proteins as well. So when during development of, for example, a chicken or a cow, there are proteins that are stored within those components, so that when that embryo or small calf is growing, that they have all of the amino acids and ability to build the proteins they'll need to grow and to be healthy and survive. Ovalbumin, for example, is a protein that has all of the essential amino acids that would be needed to rebuild up a chicken from a small egg. So these are just small amino acids that are going to be for the embryo so that it can create all of the enzymes it needs. The ferrin protein is a protein, for example, in the liver that stores iron for different processes and for our blood, for example. And casein is an amino acid in milks that it stores amino acids in milk for cows. Signaling proteins is another class of proteins, and they'll carry signals throughout the cell and allow different cells to interact and to respond to external signals. Hormonal growth factors, for example, are signaling molecules. Insulin, glucagon, those are signaling molecules that activate different enzymatic pathways. Netrin is a growth factor for nerve cells that help to generate axon growth. So there are all these different types of proteins that can be involved in regulating and responding to the environment. Receptor proteins are the proteins kind of on the front end of that reception, and they're going to be detecting signals that help send those to the further signaling molecules. So these are receptors. Examples might be like rhodopsin is a signaling protein that actually detects light and will respond to light. Acetylcholine is the receptor that's going to respond to and generate muscle contraction. The regulatory proteins are typically on that far end, are typically on the back end of that signal cascade, and they'll be expressed or activated to interact with the DNA, for example, to activate the translation, the creation of proteins from these different, from our genes, from our DNA. And again, you could have something that's activating protein synthesis or silencing that. And then there are also always special purpose proteins from different types of organisms. An antifreeze protein, for example, are found in Arctic fish and sometimes different types of clams or mussels that might be in colder water. Green fluorescent protein is a fluorescent protein that's found in jellyfish. We have various different types of monolin, which is a sweet tasting berry that is from the serendipity berry in Africa. This is an example shown here. So there are certain things that are going to be useful for different types of organisms. And another one's this glue protein that helps mussels attach to rocks. There are all these different types of proteins that are interesting and different and have unique properties that are not found everywhere, but different types of organisms will have them. All right, proteins, what are they? Well, they're made up of what are called amino acids. And amino acids contain two main functional groups, an amine group and an acid group, a carboxylic acid group. They have this general structure, all of them do. They have a nitrogen, carbon, carbon. And the middle carbon between the amine and the carboxylic acid is going to have a side chain. And we'll talk about some of those side chains and what they look like and what they are. So this is the overall general subunit. So this is the monomer subunit of a chain. So we grow this, we kind of combine these groups together into big long chains, and then the side chains give them different properties, and we'll talk about that. One of the important parts of this, and we spoke about this when we were talking about sugars, is you'll notice this carbon is a chiral center, which means that this carbon is attached to 4 different and unique things. So because of that, this is going to have special properties in terms of what we are going to be able to do with it and how our enzymes are going to interact with it. We have 20 kinds of amino acids that are used in proteins, and we're going to talk about those different groups and what they look like. This is just kind of a different orientation. So now the carboxylic acid's on the top and the amines still on the bottom or on the left. The side chain is what's going to change between these these 20 different amino acids. And again, sometimes this chiral carbon is called the alpha carbon. So here are a list of some of the amino acids. There are 9 amino acids that have nonpolar or hydrophobic R groups. So these R groups are shown here that are kind of highlighted. You'll notice that these are all of the hydrophobic or lipophilic, right? So hydrophobic means afraid of water. Lipophilic means liking fat. So these would all count for those types of non-polar side chain groups. These are the groups that are going to give these special, different, unique characteristics. Here are polar hydrophilic side chain groups, or R groups. We can see serine, threonine, cysteine. All of these have different polar functional groups that, or they're charged. So these on top are uncharged polar groups, and then the bottom ones will actually, at physiological pHs, those will have charges associated with them. So here's a carboxylic acid functional group. You can see it lost. So this is in its conjugate base form. Here we have different amines here, and they have added the protons. So these are the conjugate acid form of those functional groups. Polar amino acids tend to be found on the surface of the proteins, on the outside, facing the maybe cytoplasm or the liquid water-based contents of the cell on the outside. I want you to be aware that there are 3 different ways that biochemists use to show what the amino acid is that we're talking about. They can use their full name of the amino acid that are listed here in alphabetical order. You can have a 3-letter abbreviation that are listed here. And then there's also a 1-letter abbreviation. So 3 different ways that these are commonly used to provide information about what the amino acid is. So this is a good handy list to have available in case you're looking at protein structures or you need to know what type of protein we're talking about. Some of the side chains I mentioned and I showed you they have different ionizable side chains. So at different concentrations of acid, so depending on the physiological pH, we're going to have these side chains have a charge on them. And this is an important kind of idea that some of these will be charged and not. It's going to change the intermolecular forces that might be able to be involved with the side chain interactions. For example, these amine side chains that at physiological pHs would be positively charged, we'll notice that if we have a carboxylic acid, right, we're going to be able to have intermolecular forces that are ion interactions, and those are very strong intermolecular forces that would be possible. I don't want to spend a ton of time on this idea of the Henderson-Hasselbalch equation, but it's really an interesting thing that's been used to have a connection between the pH and the ionizability of those side chains. So basically this is telling us there's a little bit of a buffer region here for the side chains and where they're going to be and what the pH is going to be at which they are going to be ionized. So here's an example of the relationship between the pKa that we were speaking about a few lectures ago in terms of the acids and bases and the strength of those acids and bases and the pH of that, and if we're going to be in the acid form or the salt form, for example. And this is just kind of a Henderson-Hasselbalch. This is going to be a chart where we're looking at different amounts of base that are being added to a system, and then the pH that's climbing as we add that base. And then we're looking at the different orientations of the different ionizability of these groups depending on that pKa value. So you'll notice that one of the first acids that is going to be reacting is this carboxylic acid group. And that is going to start to react around a pH of 3. And then the next group that's going to come off around the pH of 5, for example, is this second carboxylic acid group. That's the side chain. And then the third one is going to be the different amine that you can see here that's positively charged. It lost a proton. So we have 3 different chemical reactions that are happening depending on the pH that's basically acting, if this is going to be able to act as an acid or not. So this is the idea of the Henderson-Hasselbalch equation. And just to kind of highlight that, I just wanted to show you that that is a difference here that's looking at these three different chemical reactions at the pH at which they're going to react. So physiological pH is around 7. So that's what some of those amino acids on the previous page are assuming, that we're working in a pH of physiological pH around 7. What that also means is the backbone of the amino acids at physiological pHs, because the pKa of the amine group is 9 and the pKa of this carboxylic acid group is a 2, typically at a pH of 7, these are going to be ionized. So this is going to be a positively charged nitrogen, and this is going to be a negatively charged carboxylic acid group. And the name for that is called a Zwitter ion, which means that it's both ions at once. So these have unique properties that allow them to have really high melting points, really high boiling points. They're very water soluble because of these properties. And it's just something to recognize as we kind of continue to talk about these. In the next few slides, I want to go over some of the basic ideas and basic components of amino acids, and then talk to you about some of the unique different side chains that we can kind of see in some of these. So first of all, some of the terminology. So this is the alpha carboxyl group that we are going to start typically with the naming of this compound. If we're looking at organic chemistry, for example, this alpha carbon is not the alpha carboxyl, but the alpha carbon is the carbon that is the chiral carbon as well, as we're attached to a nitrogen. What we're not showing you is kind of going to the back of the plane is a hydrogen that again is just inferred, but it is not being shown here. There's also a hydrogen back there going into the back of the screen. We have a functional group up here. And so this is attached to 4 different and unique things. So that's the chiral center, also known as the alpha carbon. The alpha amine is this amine, which is kind of part of that backbone structure. So the R group in this instance is shown for all of these kind of to the left of the main backbone structure of the amino acid. These are all aromatic carbons or aromatic groups that are the side chains. Tryptophan, phenylalanine, and tyrosine all have these aromatic structures. So you'll notice that they are these 6-membered rings that have double bonds in every other bond. And these are called aromatic structures because of their unique properties that they have due to that double bonded and that delocalized p orbitals. So again, the amino acid backbone is shown to the right, and then the R groups are shown in these little blue boxes here. So these are the physiological pHs at the 7, about approximately 7. You'll notice that the R groups don't change their pH. They don't get charged. They don't act as acids or bases, but the backbone, the oxygen on the carboxylic acid, and the amine, those all will change their pHs. The tyrosine could potentially ionize or release that, but you'd have to have a really high basic conditions in order to do that. These are all aliphatic side chains, or groups that like to have more of like those fats, for example. And here we can see that those side chains are mostly nonpolar. And these are at physiological pHs. These side chains don't really change. It's only the backbone, carboxylic acid and amine groups that are going to change. Now we're on to the carboxyl side chains. The R groups here have carboxylic acid functional side chains. Those include aspartic acid and glutamic acid. And these are the side chains that are shown in the blue boxes. Physiological pHs, we'll notice that these side chains will also be able to act as acids and release those protons. Here is the form at physiological pHs where now it's going to be in its conjugate base form. Hydroxyl side chains, where we have alcohol groups These are going to serine, threonine, and tyrosine all have those alcohol functional groups. We're also, and you can see those highlighted there, the tyrosine can ionize really at high pHs, but otherwise these other ones do not ionize, typically under normal physiological conditions. This is a sulfhydryl R group. It's called cysteine, and we can kind of see that side chain. This ionizes at about a pH of 8. So that's a little bit outside of what normal physiological pHs would be, but obviously it could happen, and we could ionize that at once we get to that higher pH. Of course, the side chains are typically also going to be ionized at that point as well. And we also now have carboxamide, so that amide functional group that is shown here. These are asparagine and glutamine are those groups with those side chains, those R groups. And these are going to, at normal physiological conditions, they're not going to be ionized or charged in any way. We have arginine, lysine, and histidine, and these are ionizable amine side chain groups. So if we look at those side chains in blue again, we can look at physiological conditions. We're going to have positive charges on all of those groups. So these arginine, lysine, and histidine can all get charged under normal physiological conditions. There are other non-protein amino acids like carnitine. You might have also citrulline, ornithine, taurine. You might have heard of taurine in Red Bull for energy drinks. So some of these things can be used for energy in our bodies, but they're not essential. We don't need them to survive. So they're not components of proteins that we need for building blocks, or some of them we can synthesize in our bodies ourselves. So some of these they're still amino acids because they have an amine group and a carboxylic acid group, but they're not the type that we use within our bodies for building our proteins, for example. So we're going to talk about this a little bit more as well, but when we start looking at the structure, when we create these things, there is going to be a difference in how they interact with each other depending on the pH of the system, the temperature of the system. All of these things are going to affect those intermolecular forces. One of the things that when we eat food, we have different types of enzymes that are going to break down some of these types of proteins into smaller bits. And pepsin is an enzyme that works in our stomachs. So this is really functions well at very low pHs. And you can kind of see that activity on the y-axis versus the pH. As the pH is really low, which means that there's a lot of protons in the solution. It's a very acidic environment. This is where the activity is at its highest. As we get out of the stomach and perhaps down into the larger intestine when the pH drops, then we start to lose activity of pepsin. Trypsin is in the small intestine, and that's where the pH is a lot higher there. And you can kind of see that pepsin, as that dies off, we get another enzyme that is able to cut up proteins at a pH of about 6, where pepsin's activity is maybe 25%. Trypsin's is at 100%, much better. So we're able to have these different types of enzymes that have different capacity to work at different pHs. So we talk about physiological pH within our cells and within our bodies, but obviously there are different compartments within our bodies where these conditions might change slightly, and then we can generally create different systems or different setups for those proteins to function in different environments. Some of these components can also get post-translational modifications, which means once we synthesize the protein and create it in our bodies, we can add on different compounds to those. So here's an example of a hydroxylation. So we'll add that hydroxide group onto a side chain. We might have a carboxylation, or we might have the addition of a glucose molecule or a sugar moiety onto there. So just some examples of things that can happen and can modify and modulate the protein and the activity of that protein within our bodies. Phosphorylation is also a common mechanism for regulating a protein and its function. You can kind of see here that we have a serine that usually is an OH, just an alcohol group, but now it's been phosphorylated here on the end. So this is a phosphoserine, phosphotyrosine, phosphothreonine. These three amino acids are involved and can be involved in phosphorylation events that activate and modulate the structure and the function of the protein. So once these are phosphorylated, that clearly could change some of the interactions that these side chains can make. All right, just some quick questions to kind of see where you're at in terms of understanding what the components are and what's the side chain versus the backbone of these amino acids. What the amino acid shown below has a side chain that is what? So this is going to be a threonine, and is it acidic, basic, nonpolar, or polar? So pause and think about this, and then we'll talk about the answer. All right, so we're gonna look at this, and it's polar and neutral. You're like, wait a minute, there's charges on here. Remember, this is the amino acid has a side chain, so we have to be able to distinguish between the side chain and the backbone. This NCC, this is the backbone of the amino acid, and this group down here is the side chain. So it's polar because it has this hydroxide, this has this alcohol functional group, but it's neutral because it's not charged at a physiological pH. All right, the amino acid shown here has a side chain that is, and what of these would be the answer? So pause and look at the structure and decide what you think the answer might be. This is a little bit of a trick question, so I'll just warn you there. The answer is actually E. It is hydrophilic because this is a very polar group and it would like to be by water, but an amide functional group is typically not very basic. And it's because that positive end of the carbonyl group attracts the unshared pair of electrons on the nitrogen strongly enough so that it actually prevents it acting as a base and accepting a hydrogen atom, for example. So typically, as a result, amide functional groups are not basic like amines are, like this amine functional group here on the backbone of that structure. Which of the following amino acids can form hydrogen bonds between their side chain groups? This one I'm not going to answer for you. I would like you to post your answer for this question in our Blackboard space. Now that you've been introduced to the amino acids, we're going to start to talk about how they fit together and how they go together. And you'll notice that here we have a glycine. Here is the N, the C, and a glycine, the side chain is just a hydrogen. So we don't have to really, there's not really much of a difference there. This actually isn't even a chiral carbon because of that, this alpha carbon. But we're going to attach this to an alanine, which is also a pretty simple side chain group of just a methyl group. What is actually happening is we're dehydrating this compound. We're going to attach this carbon to that nitrogen here, and you'll be able to see that is going to form a peptide bond. So we're actually going to eliminate this water. So we're dehydrating this in the water's forming here, and we're going to make a new bond between the carbon and the nitrogen. And the other thing that's going to happen is then we're going to form, it's called a peptide bond. We're also forming, we're creating this amide functional group. So this is a glycine alanine combination now. You'll notice that there's a couple of different things. We have a directionality. So we have the carbon and the nitrogen attached to each other. We don't have nitrogen and nitrogen or the carboxylic acid. It's these two groups that are forming the amide. So there's directionality, there's that peptide bond that's forming, and we'd call this a peptide until it's folded. And then once it's folded, then we can call it a protein. This is what a peptide looks like once we start adding more units of our amino acids. So here we have a methionine, we have aspartic acid, leucine, and tyrosine. You can see the names down here with the 3-letter designators. And the backbone is NCC, 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 NCC. There's directionality. We have the amino terminus, more often referred to as the N terminus, and the carboxyl terminus, more often referred to more often referred to as the C-terminus. So the N and the C-terminus. And here is the side chains are going to kind of come off to the sides and out on either side of this. Here is the peptide backbone, or that amide functional group at each of those locations. And we can continue to add on and extend this chain. We're going to always have directionality. So we start with an N-terminus. When we synthesize these, and I know we haven't talked about this, but when we create these in our cells, we're generating this information to create which one of these we're going to put in as dictated by our nucleic acid sequence, or our DNA sequence. And our DNA sequence is going to dictate which order these go into. And we're always going to make it from the N-terminus is going to be the start. And we're going to end, we're going to synthesize it. The C-terminus is going to be always at the end of the peptide or the protein chain. Here's a cartoon representation of a peptide as in the unfolded form. And when we have something that might be polar side chains in blue or yellow, and we have green would be non-polar side chains. So typically what's going to happen as this is being synthesized, these different groups, especially the non-polar groups, are going to kind of glob together, kind of like oil and water, right? Those are going to be pulled together, and we're going to create this globular structure that is going to have these more charged compounds or side groups pointing out, so that they can have intermolecular force interactions or hydrogen bonding with some of the water in the outside surface. And so this is kind of the more folded versus the unfolded peptide versus a folded protein. So these different types of intermolecular forces help to shape and organize the structure. And remember, we're going to have the van der Waals forces in the middle. We're going to have stronger intermolecular forces typically on the outside. Cysteine is a unique structure, and what happens when some of these get together in a protein, we can actually make these disulfide bonds. So if you have a cysteine as shown here, sometimes what they'll do is they'll interact with another cysteine within the chain, and you'll get a compound that would be close to it, and then we're going to have that side chain. What will actually happen is we can make that side chain will bond, and we'll make what is called the disulfide bond. So why is cysteine residues with a free sulfhydryl group tend to localize on the exterior? Well, this is going to be more polar, more electronegative, than the hydrogen. So there's a polar difference there. But when we have a disulfide bond, these are going to be sharing the electrons in this bond evenly, so there's not going to be a polarity difference anymore. So now this new disulfide bond, this is no longer a polar side chain. After that, hydrogen has been removed, and so then that will typically get buried in the inside of the molecule, and it could definitely change the structure from if that's free versus if there's a disulfide bond present. So we can do different things, and some experiments that have been done to look at protein structure can help to identify and understand how these things are folded. Urea, for example, is this structure right here. It's amine groups with that carboxylic acid, or it's this amide functional group with extra nitrogen on the other side. And this can sort of kind of interact in a similar situation or a similar kind of way that the backbone, it almost looks like an amino acid, but it's a little different. And it can get in and intercalate in and disrupt some of the intermolecular forces, and it can help denature a protein. But if you remove the urea, it'll reform typically back to its original state. So there is something that is innate in that sequence of protein of the amino acid, and the peptide sequence is going to dictate how it folds and what it is going to be looking like and its structure. There are also in our cells and in our bodies different ways that we can help to regulate the shape through proteins called chaperone proteins, or different types of heat shock proteins, which where either they help to fold around, or you can create a more kind of contained environment that has no water, for example, or no high salt concentrations that will help to correctly fold the protein of interest. Sometimes proteins can act as their own kind of mechanism for these kind of, in a similar way as the chaperone protein. Sometimes proteins can aggregate and cause problems. So this is an example of maybe in a protein, an amyloid fibril protein. Amyloid is something that can happen in Alzheimer's disease, for example, or someone with brain trauma, brain injury. We can get the misfolding of a protein, and that misfolded protein can recruit normally folded proteins and then activate it and convert it into an abnormal folded protein. These are called prions, and prions, you might have also heard of chronic wasting disease, Creutzfeldt-Jakob disease. There are all these brain diseases that are related to these prions, and they can be dangerous. They're also really dangerous because it's hard to clean them up. So in terms of a food safety kind of situation, these are very, very resistant. They're typically, we'll talk about this in a little bit, they're typically a type of secondary structure that we're going to talk about, these flat, very stable proteins, and they're really hard, even with high heat or high salts or acids or bases. They're very resistant to being cleaned up. And so it's kind of a very big concern that if a food has a prion, or an animal has prion disease, and you were to use that in a food processing facility, you could potentially be passing on these prions. So it's important that we test for these things and know that they're not, don't contain these. So that's the concern with chronic wasting disease. Right now, we don't think that it necessarily passes from, for example, deer to human, but if people don't want to take a chance on that. So it's very important that we're doing the testings that we do for CWD and making sure that spoiled meat that has these prions in it don't get eaten by people, so that then there wouldn't be some type of cross-contamination and other issues that would arise from that, consuming that meat. There are several different types and kinds of intermolecular forces that regulate and dictate the folding that's going to happen of these proteins as they're synthesized. I mentioned that we can have covalent bonds, and one that I showed you a few slides back was the disulfide bond. So these are pretty rare, but when we have cysteines that can be close to each other in that sequence, we can create these disulfide bonds, which kind of folds and maintains this folded form very well. In the purple is just the backbone, that NCC kind of backbone structure. We can have hydrogen bonding if those side chains have that capacity. We also could have ionic bonding depending on the side chains, if they can be ions of different types. And then we also have the van der Waals forces that are going to be involved in some of those side chains that are just carbon hydrogen based without any polar groups. So these are some of the main different types of intermolecular forces that are going to dictate the shape and the folding pattern of the protein. I'd like you to write this out and post this in our Blackboard space. A couple of different things, 4 different types of peptides. So a peptide that would interact with water, a 4 amino acid peptide that would be associated with a lipid bilayer, so more of a fatty type of peptide, a 4 amino acid peptide that might interact with a negatively charged metabolite or component, and identify and draw a pair of amino acids that would interact via an ionic intermolecular force. So 4 different peptides or 4 different groups, and post this in our Blackboard space as a little bit of practice working with these structures. So I'm going to stop here now that we've kind of gone through some of the basics for amino acids and how they fit together. The next part, we're going to talk more about proteins and protein folding and how that kind of relates to each other. And then we'll continue on with that in that next video series, that next video lecture.